This is the first of three parts about the narrow escape time, and I will give today a general introduction about the subject. So the narrow escape time basically corresponds to rare events in physics and biology where a particle has to find a small target. So this general introduction is about where this problem occurs in physics, chemistry, virology, biology in general. And there are two more lectures that you will see also online, Narrow Escape Part 1 and Narrow Escape Part 2, which corresponds to the mathematical part. There I will develop the methods coming from probability, analysis of partial differential equation of Laplace equation type, to give an asymptotic formula that we will see also today, but not the proof. The proof will be done there. So the general overview is the following. Really this is about the motivation of this time, about how to find a target. So we will see the first example is about the nucleus of a cell, which contains the genetic information. Another example is synaptic transmission, which is the phenomenon occurring at the junction between two neurons of the nervous system, for example. Another example that we will discuss is the process of motion of receptors, which are channels protein occurring on the surface of neuron. And the last example will be coming from the process of transduction, and specifically about phototransduction. Then I will spend briefly sometimes to discuss the mathematical formulation of the problem and how this is important for coarse graining of simulations. So let's start with the cells, which is really what we are interested in. And this is a typical cell. This is a schematic representation where you have the boundary of the cell. You have here the nucleus, where the genetic information is located. And if you think, for example, of what is the process of a viral infection at a single particle level, you have a virus that can enter here. So you suppose you have a virus and it enters here, whatever the mechanism. And what it has to do, these viruses has to, for some cases, I'm speaking generically, it has to go for some of them to find a nuclear pore, which is a small portion here of the nucleus. This is called nuclear pore, where you have these small doors where when you find it, a particle such as a virus or the RNA or the DNA can be translocated inside. So once you are inside, so this is here the nuclear pore. And another question about a small hole question is, suppose you have somewhere here a part of the DNA which contains a site, and you are asking for how long it takes for a factor which is called transcription factor, which is denoted TF. This is a protein, a molecules that moves by Brownian motion, and it can move, and this is, we will discuss it in a future lecture. It can also move along the DNA. If you have a piece of a DNA like this, it can also move on this DNA and then get detached. And we're asking how long it takes for this to find here this small target. So this is a target site. So again, the transcription factor can scan quite a lot about the nucleus and the DNA. And still, it has to find a small site where it, upon binding, will generate, for example, a messenger RNA, which is important later on for protein synthesis. Okay, so let's continue our general introduction. So another example which is quite important for synaptic communication, neuronal communication, it's a domain which is called a dendritic spine. Here you see several examples of them. You see this is like a protrusion. This is a dendrite that you can see in here. So let me change the color. So here in red you have the dendrite of a neuron. And what is quite interesting is that you have this protrusion here that have different shapes, and protein, or if we think about the process of transduction, so you have here an example in yellow of such a structure. When a vesicle, that you see this little dot here, let me magnify this, you see this little round that you can see inside, that upon release of a vesicles, some channels open, we will see that later on, and then a current of ion enters here, such as calcium ion. You can have, let's say, something like 3,000 ions that enters into a single NMDA receptor. This is a type of receptor that lets calcium goes in, and then calcium move by diffusion, and they have to escape such a structure. And an important question is how long it takes for this to happen. And so this falls again into a narrow escape problem, because once you're in here, the ions, they have to find here a small exit. They have to find a small target to exit all along and then reach the dendrite. So here's more specifically the way the spine can be modeled. It can be modeled as a round head connecting a narrow neck. So this is the neck and this is the head. So you can either connect smoothly here with a structure which is of the shape of a cusp, or you can connect in a discontinuous manner, just putting a cylinder here directly to the round head. So if you think of a particle that enters here in the 3D, then it has to move and find here this small opening, or in here again it has to find a small opening. And then the question is, of course, what makes the difference between this type of connection, let's call it connection 1, and the other type of connection, which is connection 2. And this is a problem that you can find in the literature under the name of Dire Strait time. So let's discuss more specifically the process of synaptic transmission. So again, this is an example of a synapse which does not occur on the dendritic shaft here. This is called the dendritic shaft, but it occurs on a dendritic spine, which is here in yellow. And this is called the presynaptic terminal that you can see in here. And the spine is called the postsynaptic terminal. And upon an action potential, so the sequence of event is that one, you have an action potential, AP. Then the action potential leads to some calcium flows here. And then, so calcium flows into the presynaptic terminal. Then you have this induces vesicular release, that is a vesicle containing neurotransmitter, is going to fuse, and you see this small little point which representing neurotransmitter. Then the neurotransmitter, they are diffusing inside the space in between, which is called the synaptic cleft, that you can see in here in blue. So this is called, this region here in the middle is called the synaptic, the cleft. And again, this molecule, about 3,000 of this molecule, they have to find these receptors which are located here, which are really representing, I will for clarity put them in red, so this is exaggerated, but it's to show that there are actually some of them, and they have the molecule, they have to find these receptors which are really small targets. And this is the receptor here. This is an example of AMPA receptor, which is a transmembrane protein going back and forth inside the membrane. And this is an example of the NMDA. Again, this is a schematic picture, and you see from the NMDA calcium can go in. And why this is important, so let me continue the sequence. The neurotransmitter, they find the receptor, so they activate the receptors. And once the receptors are activated, there is a flow of ions. And really where the narrow escape, finding a small hole occurs, is when the neurotransmitter are released and are activating this receptor. So this is in between 4 and 5 in these steps. So again, one interesting question would be how long it takes for neurotransmitter to find the receptors, how many receptors are going to be activated, and how this depends on the geometry of a synapse. So this is again more specifically all the events that occur when a vesicle is released, and you can see here the location of the receptors in red. And interestingly, they are holding, what hold there is some structure which is called the postsynaptic density, PSD, post-synaptic density. So you have here dense structure made of proteins, that's the name of those proteins are called scaffolding protein, that are maintaining these receptors. And now, of course, you would like to derive the currents that will be generated inside here, the postsynaptic terminal, and how this current depends, one, on the number of receptor, which is here n of k. Gamma k is the conductivity, that is the amount of ions that enter through a receptor when it is open, and p is the probability that a receptor located at position x, and the type of receptor is labeled by k, this is the probability that this will open. And dV is the difference of potential. We assume here that V is not going to vary while the current is entering, which is not necessarily the case. This requires for the full expression to solve what is called the PNP equation, the Poisson-Nernst-Planck, which I'm not going to discuss here. Poisson-Nernst-Planck. Right? So now what has been found in the last 10 years or so is that the number of receptor is not is well regulated. First of all, receptors are not fixed, but they are moving. We will see that in a second. And what we've been able to calculate, which we will show in next classes, is this probability. What's the probability for a receptor K at position X to open at time t? Basically, this corresponds to calculating what's the probability for a particle that moves and find here this receptor, and this has to be compared to the other type of trajectories that are going to escape here, and once they escape it's very difficult for them to go back. So this arrow of going back is almost negligible. All right. So this problem, for example, has been addressed using Brownian simulations, and it is quite challenging to design correct simulations and to account for the stochastic effect of each component, such as the opening or the closing of the channel, that they can also be in another state which is called the desensitized state. You have to account for the position where vesicles are going to be released. You have to account for the number of receptors that you are going to place at the postsynaptic density. You can also play with the geometry. So the simplest example of a geometry is to put a cylindrical type geometry where you have symmetric cylinders which are symmetrical along this line in red. And if you want to account also for other type of cells that play an important role, you have also to use glial cells and put what are called transporters here in blue. And the role of the transporters is the following. Once you have a Brownian neurotransmitter, it can of course find and bind here to a receptor, but it can also bind here to a transporter, and in that case it is transported here inside the cell. So this here corresponds to an astrocyte. So those are what is called glial cells. So we and other groups have derived a model for such a simulation. So if you are interested, you can have a look at this publication in PLOS ONE by the first author is Dominic Fresh, where this is exactly what we implemented. In 3D you have here the cylinders, you place here the receptors that can eventually move by Brownian motion on the two-dimensional surface. Vesicles are released, so they are shown here these are the blue dots here. Those are the neurotransmitters. And they can bind here to the green receptors. And again, this is a small hole problem because the neurotransmitter have to find these small targets. Now let's discuss another aspect where we're going to find the narrow escape problem is about trafficking of receptor on cellular membrane. So we are still looking at neuronal cells, and this is the presynaptic terminal and this is the postsynaptic terminal. Now in the last 15 years, people have discovered that the number of receptors that are involved in synaptic transmission is of the order of a few. So here you have an example where you have 1, 2, 3, 4, 5, and 6 NMDA receptor located at a single synapse. And here this is a view from above where you have in blue the NMDA receptors, in white the AMPA receptor. Those are the two glutamatergic receptors. And you can see that they are of order of a few, and this was important because it was important to understand this because this shows that you need to have a stochastic approach because you are going to have a fluctuation with a small number of particles of receptors. And one important question is, of course, how receptors are going to find their correct location. They have to move on the surface of the neuron. And this motion has been described as Brownian motion. And you can see here an example of a trajectory of a single receptor. And in blue, you can see a type of free Brownian motion. free Brownian motion, and in red you have the confined one. So this is what you see in red, which is confined Brownian motion, and in green this is a label that you are sure that you are at a synapse. This is a labeled of the PSD, of the post-synaptic density, which I'll call P, S, and D. And it has been shown that receptors can alternate between free and confined Brownian motion. As you can see here, the succession of blue, blue, red, red, blue, red. So this succession says that the particle moves with this different type of motions. And this graph represents the expression of the mean square displacement, where people have plotted the position at time t plus delta t minus the position at time t, and they average this squared, and they show that it behaves like it's proportional to time, with a coefficient here which is really proportional to the diffusion coefficient. Depends here, the c depends on the dimension. So we have seen that if they move this way, so you have here another example of this type of motion, from very confined 1, less confined 2, even less confined 3, not confined at all 4, and 5 is the switch between confined and not confined. Interestingly, if you are interested in how this motion correlates with the underlying geometry, with the underlying surface of the neuron, then this is what people have reported, that you have a Brownian motion which can only occupy a small piece, a small part of the postsynaptic density that we have discussed previously. So you have the explored space compared to the unexplored space, and yet the receptor have to find their correct target, which is again a small hole. So we are again facing a narrow escape time question, which can be formulated as you have here your target, and you have a Brownian particle somewhere, and you have restriction of the geometry, and this target can, for example, be on the surface of the PSD or inside, and yet the particle has to find here this target. And the question is how this depends on the geometry and other physical parameters. So to clarify the role of the geometry, it's important to realize that the membrane, the surface membrane, is very crowded. You can see here example of proteins. You have here this this line are the filaments. So there are filaments, proteins, and what is interesting is that all of this can generate domains that are represented here schematically. You see, this is, I'll show it in red. You can here have this domain delimited by these yellow tubes. So those are confined microdomains or even nanodomains. So to address the question of crowding, such as you can see here in the cytoplasm of the cell, where you see that really the medium is very crowded, it's very hard for a Brownian particle, which we are going to represent here in blue. If you have a Brownian particle in here, it's very hard for this particle to move and to find its way in this very dense medium. So this can be analyzed using a model where you can place obstacles on a lattice. This is an example of a square lattice. So on this lattice, if you want to put a particle, a Brownian particle somewhere, the particle can move. And then what you can change to explore all the possibilities is the size A of these orange obstacles. That when the particle hit here the orange places, it is reflected. And then of course, when it arrives here at the end of the square, it moves in another fundamental domain, in another domain. So the vision of how obstacles can affect diffusion is the following. You can see diffusion at two scales. There is the first scale, which is the scale inside this domain here, which is basically the time it takes to escape. And this gives you one scale of diffusion. This is a small scale. So this is the scale inside the domain, which we'll call the where you have the real diffusion. But this is not what you are necessarily observing under the microscope, because this is usually inaccessible. So what you see is, you see jumps. You see that there is, the particle goes from this to another square like this, where you have obstacles, and what you see is really diffusion at a coarse graining level, where you go from one square to another square, and this is called the coarse grained diffusion. And this coarse grained diffusion is usually what is observed. And what is interesting is, if you know this, how can you recover from this? How can you recover the density of obstacles? So this problem falls precisely on the type of narrow escape domain, because you have to estimate, to calculate the time it takes to reach here this edge, this free edge, this opening edge. There are four of them, of the fundamental squares. So I give here some references, classical references about the effect of crowding, starting from papers in the 80s. Then in the 90s, and there was this fundamental work of Kuzumi about visualization of the 3D membrane. Then the theoretical work of Zach Stone, which gave fundamental principles for anomalous diffusion and diffusion in general. And we summarized properties of diffusion in a recent review about control of fluxes by narrow passages and hidden targets. So the next example is phototransduction. So what is phototransduction? Usually in the retina there are two types of photoreceptors, which are called rods and cones because of the geometrical shape. So these cells can be divided into three parts. The outer segment, which is the part where the transduction occurred. The inner segment, which contains the cell body and a lot of nutriment for the cell. And then the synaptic terminal, which will transmit the information to the next layer of cells. You can have here bipolar cells, and this is an important part of the transduction pathway in the retina. So what is the mechanism of transduction, of transforming a photon into an electrical signal? So this has been well studied for the last 30-40 years, especially by Hodgkin and many of his followers. And it goes as follows. You have a photon, and when the photon hits a molecule which is called a rhodopsin, the rhodopsin becomes activated. And I'm not going to here to discuss the entire cascade of chemical reaction, but basically it activates G protein molecules, and ultimately a phosphodiesterase molecule here, the role of which is going to hydrolyze cyclic GMP, which is an important molecule that trigger the opening or the closing of the channels. That is, when there are no more cyclic GMP, the channel close, and if they are closing, all the calciums and the ions are going to be extruded here through the exchanger, which will lead to an hyperpolarization of the cell. So where is here the small hole problem? Well, we have to understand that in darkness, when there is no rhodopsin activated, we calculated that there are in average only one phosphodiesterase which is activated, which we are going to call PDE star. And this molecule, which represents a small target, is enough for to balance the generation, the production of cyclic GMP. And cyclic GMP molecule, they have to move and find here a small target, which is here inside a compartment. A compartment is the space in between two discs, and you can see that in the outer segment compartment contains about 1,000 of these discs. So each of these molecules that will hit this PD are going to be hydrolyzed, and this what maintains a fixed, I mean, it is not really fixed, it is fluctuating, but it is fluctuating above a certain level, and this level defines what is called the dark noise. And it is possible to estimate the rate or the time it takes for a particle, for a molecule here to find here this target as a function of the geometry, which is here A is the size of the target, L is the length of a compartment, and R is the radius of the compartment. So using this, it is possible to estimate the rate, which has been derived in a paper written by Jürgen Reingruber and myself in 2011 in Siam Journal of Multiscale Modeling. Multiscale modeling. And what was interesting is that we could, by using the parameter, we could calculate what is the encounter rate, which is this rate here, in case of toad and in case of mouse rod. So finding a target in the nucleus, this is what we have started to discuss. So again, the nucleus is still a mysterious compartment which contains the DNA. The DNA is folded into a multiscale structure which is called the chromatin. And this artistic concept about having chromosome floating around is still not clearly understood, how the chromosomes are organized. So we've discussed that when a part of the genome is activated by a transcription factor, this falls into the class of narrow escape problem, because you have to find a small target. Another problem which is quite important is the problem of looping. That is, you have the DNA here and you have a promoter site, and if by looping, that is by just fluctuation of the shape of the chromosome, you can bring a repressor, a protein that will bind here, it can prevent transcription to occur. And this is here the example of the lac operon. For the lac repressor, it can just block transcription. And of course the question is, how frequent are those loops? You see, suppose you have a polymer which can be modeled as bead connected by spring. So you can ask how long it takes for the two endpoints to come in close proximity, let's say in a radius epsilon, where epsilon is small. And how this depends, for example, on the length here between A and B and the properties of the mechanical properties of the polymer. And again, so this is exactly what we discuss. What is the frequency of loops? Because if by looping you can prevent transcription, this is an endogenous way of regulating transcription. So a first approach that we started for this is to imagine that you have rigid rod, and this is just to give you an idea of what is the type of modeling that you need here. And so you start, let's say, at a given position, and depending on the number, so you have, let's say, position R1, R2, R3, R4, and so the position Ri can be written as a position of Ri minus 1 as a certain length b exponential iti, where I here represents the complex number. And so the last position is just the sum over the different position rk, and theta is just a Brownian variable. So what is the meaning of a loop? A loop, if you fix, let's say, the first one, R1, as the origin, then you are really interested in when the last bead, which is here Rn, will be at a distance of epsilon from the center. And this defined actually your small hole problem, except that here you are in high dimensional space, because you are in the space of defined by all of the bead R1 to Rn, which is, if we are in dimension 2, this is 2 multiplied by the number of bead, which is 2n. So what is the shape of the domain we just discussed? That is the ensemble of angle that will give the 2 points to come into close proximity. So let me redo it. So you have 0, you have your polymer, and we are interested in what are all the possible angles, the possible location of all rk, such that the last one, rk, is in close proximity to r0. It is less than epsilon. So if you have 4, in the case of when we have 4 beads, this is what the boundary of the space look like. That is, if you have particles in this space which represent the position of rk, then rk will be in a position where it is less than epsilon of r0 only if it is located here on the red structure here. So this is the boundary, and you can see that the boundary is quite complicated. There are some portion of blobs which are connected by small narrow tubes, and you can see here a simulation of a Brownian particle in this high dimensional space that represent the configuration of a polymer evolving to the one which is going to be such that rk is in the neighborhood of r0. So if you are interested in more detail, which I'm not going to give here today, you can go and look at this paper in Amitai in SIAM Multiscale Modeling and Simulation 2012. So finally, the last example I would like to discuss today is the problem for a monomer located on a polymer. And again, it falls into a narrow escape type problem, is how long it takes for this blue particle to find here the target in red, which is here a small target. So this problem is quite difficult. We could not solve it analytically. We only did a simulation. And I will leave the following reference for the detail of how this time can be calculated. So just a few words of introduction for the next two classes about the narrow escape theory. So the narrow escape theory is the methods to estimate the time for a small particle to find a target when it is Brownian. So here is an example of a trajectory of a particle, a Brownian particle, that has to find here the small target in green. And this is dimension 2. And now this is the same in dimension 3. So the mathematical analysis is to calculate this time as the size of the target epsilon. So epsilon is actually the ratio of the absorbing part of the boundary, here the green, divided by the total part of the boundary, d omega. So this is omega, this is a domain, and d omega is the boundary of the domain. And what the asymptotics gives, and again this is the subject of the next classes, is how this time depends on A, the surface, pi is 3.14, d is the diffusion coefficient, and epsilon is what we just defined here. So the mean first passage time does not depend on the location. Whether you start here, here, or here, or here, the time is the same, except if you start in a small portion of the absorbing boundary. So the next term in the expansion is of order 1 in dimension 1, in epsilon. And these depend actually on the initial position, and this can be seen through the analysis based on Green's function, which we will see in a different class. Green's function analysis. So in dimension 3, it's not anymore log of 1 over epsilon, but the time to find a small hole depends on the volume, the diffusion coefficient, and A. A is the radius here of the small absorbing disk. And then you have here the mean curvature at the center of the disk, and then the next term is log of A, right? And this should be like this, and then we have a term of order plus a term of order 1. When the narrow escape occurs not at a smooth boundary but at a cusp, surprisingly, the time it takes in dimension 2 is not log, but it's 1 over square root, which is much longer, because the particle has to find here a small hole. If you're on a manifold, and if the surface, this is a two-dimensional manifold, and here, if you know exactly what is the decay here at the cusp, which we suppose is of this type, A plus some power, this is the local Taylor expansion, the mean first passage time, so this is for a two-dimensional manifold with a cusp. It's really not important what happened, let's say, outside the cusp in this part of the manifold here. What is important is the cusp, and then the mean first passage time, as you can see here, depends on 1 on nu divided by so it's nu divided by 1 plus nu at the power 1 over A. In dimension 3, again, if A is the size of the absorbing hole, the power that you have here is tau is 1 over a, with there is a constant a to the power 3/2, which is a different power law than the 1 over a that we have seen before. And again, we have developed a methodology to derive those formula that, as you will see in a second, are important to coarse-grained Brownian simulations. Other type of mean first passage time is, suppose you have a Brownian particle moving here inside an annulus and can enter only through this hole here when it is reflected here inside the inner radius and outside the outer radius. It turns out that this is a two-parameter problem where you have the ratio of the inner to the outer radius of the annulus and epsilon, which is the size here of the small hole. And again, in dimension 2 you have log of 1 over epsilon, but you have also a term which is log of 1 over beta, which can be important. Another interesting location of the small hole is when it is at an angle. So in that case, instead of having the classical narrow escape formula in dimension 2 with pi, we have here alpha. And of course, when it is located on a flat surface, you obtain here alpha equals pi. And again, if you are in between two circles and it creates a cusp, you have this 1 over epsilon dependency for a particle here that has to find a small hole located here of size epsilon. All of this has been summarized and discussed in this publication in Zinger, Schuss, Olkman in Narrow Escape 2 and 3, published in the Journal of Statistical Physics. So what is the equation to be solved? The formulation of the problem is the following. If you have a Brownian particle, x dot equals square root of 2d w dot, where w is the Brownian motion and d is the diffusion coefficient, using the Ito calculus that we are going to see in the class, you can show that the mean first passage time, that is the time tau is the mean first passage time, this tau, which depend on the initial position, so we will write it 2 of x, which is here called u of x, satisfy a partial differential equation which is d Laplacian u equal -1, u equals 0 in the absorbing part of the boundary, du dn, which is a reflection on the reflective part of the boundary, and you would like to estimate u epsilon of x when epsilon, which is the ratio of the absorbing to the reflecting part of the boundary, is small. So this is the mathematical formulation of the problem. So I'm not going to discuss here this formulation, just to give you applications. We apply this to calculate the residence time for a Brownian particle in a narrow domain. Here this is a disk, and this was used to estimate the receptor residence time of a receptor in the postsynaptic density. So using numbers that can be found in the literature, you can estimate the time to be of the order of seconds, let's say around a minute. Interestingly, if you place an obstacles just in front of the escape, then the time is drastically increased, showing that any kind of remodeling by repositioning obstacles can lead to a really change in the residence time of receptor inside the nanodomain, such as the postsynaptic density. For example, if you even move a bit inside the receptors, you see that we can divide by 3 the time from 1000 seconds to 300 seconds. And again, when the door, the opening is completely free, you can even divide this by a factor 8 or so. So what is the final application for this is coarse graining. That is, if you have to do Brownian simulations where you have a domain and you have a narrow escape problem, instead of making a full simulation and waiting, and you know that the time is going to be long, because in dimension 2 the time, or in dimension 3 the time is going to be volume divided by a constant divided by a, by the size, by this is 4dA plus some terms. So you see that if a is small, the time is going to be very long, and so we know that the arrival time is Poissonian. So you can simply replace these entire simulations. You can replace this by just the rate of arrival, which is 1 over the mean first passage time tau, and you can give it a Poissonian rate, which is exponential lambda t. So you can inject your particle with a certain Poissonian rate instead of making the Brownian simulations. And this can be done in many places, especially if we have to simulate 3000 ions and they have to find small targets, for example here molecules. You can replace this and using Gillespie type simulations. So to conclude, we have discussed here applications and motivation really about the narrow escape problem, where it comes from in biology, what is the physics of this problem, and their asymptotic expression is quite important, because this allows to coarse grain the geometry into rate constant. And this fall into what is called the narrow escape time problem. And the formula allows to explore really the parameter space, you know, how, what is the influence of the volume, the diffusion coefficient, the size of the hole. And we've seen also that this can, once you have such number, such expression for the rate, you can use it to coarse grain and make numerical simulations. So I will stop here and give here some references, some general references about where to find analysis. So the first paper that we wrote on this was in 2004, Journal of Statistical Physics, and you can look at our literature. I'm also going to refer to the work of the group of Michael Ward from University of British Columbia, which has developed quite of in-depth methods based on matching asymptotics. So we are going to have 2 classes on mathematical derivation of a narrow escape on for smooth surface, when the target is located on the surface, and then when they are going to be a cusp. Okay, so thank you for your attention.